So, in today's lecture, we are going to look at nucleic acids, which are very important components of the functioning of a cell. So, primarily today we will look at how DNA functions, and we will try to understand the structure of DNA. Okay. So, as we have discussed previously, the DNA carries the genetic information of a cell, and what happens is that this genetic information needs to be transferred to RNA, which is then subsequently transferred to proteins, and the proteins are the workhorses of the cell. So, in order to understand the entire functioning, we need to learn a little bit about the basics of nucleic acids. Okay. So, much like proteins, which we have looked at previously, DNA also has a primary, secondary and tertiary structures. Okay. So, while proteins have about 20 building blocks, which are 20 amino acids, DNA has only 4, that is deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine, the structures of which are shown on the slide. So, if you look at the fundamental component of the DNA contains a base, which are basically, there are 2 bicyclic purines, which are adenine and guanine, and there are monocyclic pyrimidine structures, which are cytosine and thymine. The sugar is the same in all 4 nucleosides. Okay. Now, once this the nucleoside is building block is ready, the way it is joined together is through what is known as phosphate groups. So, here is the phosphate group. Here is a structure of a nucleoside, for example, the analog of thymine, and the way it is linked is through a phosphate diester. Okay. So, with 4 types of building blocks, the primary structure of DNA is far less varied compared to the primary structure of a protein. So, as a result, it was for long it was thought that DNA would have perhaps only a minor role to play in cellular biochemistry, because being such a simple molecule with only a minimum amount of variation, how is it that you can achieve complexity which is present inside the cell? This mystery is solved if one looks at the secondary structure of DNA. So, in the secondary structure of DNA was solved by Watson and Crick, which is basically a double helix. I think all of you are familiar with this concept, so I will spend very limited time on this concept, but DNA exists as 2 single strands which are bound together as a double helix. Okay. So, you have here the first strand, let us say, that runs around here, and here is the other one is the second strand. And what happens is that you have double helix has a major groove and a minor groove. Okay. And these are important when in terms of when we are looking at certain anti-cancer drugs, which are known as intercalators. We will discuss this in detail later on in the course, but what these do is that they can go and bind to the major groove or the minor groove and prevent or destabilize the DNA and prevent the cancer cell from replicating. So, also the DNA has the interaction in DNA is through hydrogen bonds. Okay. The most important characteristic of DNA is that there is pairing of nucleic acid bases. Now, what has been observed is that adenine always base pairs with thymine, and guanine always base pairs with cytosine. Okay. And the nature of interaction between these two is through hydrogen bonding. Okay. Now, between adenine and thymine there are 2 hydrogen bonds that occur, whereas between cytosine and guanine there are 3 hydrogen bonds that occur. Okay. And this allows for a constant diameter of the double helix. So, you can imagine that this bicyclic purine base, once it base pairs, you will have a constant diameter of the double helix. The double helix is also further stabilized by the fact that if you imagine that there is one base which is base pairing to the other one like this, and the next one goes this way and the next one goes that way, what happens is that these base pairs can actually have pi stacking interactions. So, here is the base here and here is the base here, and so you can imagine that they can be hydrophobic interactions which can occur, which also further stabilizes the double helix. Lastly, as we mentioned earlier, there are these phosphate groups which link the DNA, and these phosphate groups are actually highly polar in nature. So, when DNA exists in aqueous environment which has a lot of water, this polar group helps in interacting with water, and again it further stabilizes the structure. So, in summary, the DNA double helix is a very highly optimized structure wherein there is hydrogen bonding, there is pi stacking or hydrophobic interactions, and also polar interactions with water by the phosphate groups. All these together help in making DNA an extremely stable structure. An important component of the structure of DNA is that there is complementarity. That means adenine always binds to thymine and cytosine always binds to guanine, which means that if you take any one chain and look at the sequence of one chain, the other chain will always be complementary to the first chain. That means when you have guanine in the first chain, you will always have cytosine in the other chain, and when you have adenine in the first chain, you will always have thymine in the other chain. Okay. This is very useful because you have two strands which are complementary to each other, and when you have when the cell replicates or when the cell divides, you will find the first step is that the double helix unravels, and now when the new chain is constructed, the new chain uses the original or the parent chain as the template. Okay. And so, when the DNA grows, it grows and it forms the complementary base pair, and once it replicates, you have two DNA daughter helices which are identical to the parent. In other words, each of the original chain acts as a template for construction of a new and identical double helix. So, what this ensures is that during cell division, the genetic information that is present in the parent is actually transferred to the daughters. How does this process happen? What happens is that when you have a growing chain, you have a 5' end and a 3' end, and here is the growing chain. The incoming trinucleotide recognizes the base over here, and as we mentioned earlier, thymine is going to preferentially bind to adenine, and so therefore, this interaction is highly favorable, and so you have a hydrogen bonding over here, and therefore, once the base is set in the right position, a reaction can take place which can kick out the leaving group, in this case a diphosphate, and form a new phosphodiester bond. Okay. So, to summarize, what happens is that in the growing chain, the next base that comes in is always complementary to the template chain. Okay. So, the thymine always reacts with adenine and forms an intramolecular hydrogen bond, which then helps in making the or in chain elongation with a sequence specific manner. This is known as enzyme catalyzed splicing. Okay. So, to see the organic chemistry over here, what happens is that you have a phosphodiester bond, which then the alcohol in the sugar attacks the phosphodiester and kicks out a bisphosphate and produces a bisphosphate here. Okay. But you may wonder that since DNA has only 4 nucleotides, how does it transfer the information to make 20 amino acids? So, that there is some mystery over here, isn't it? So, the way this happens is that the DNA single base pair of DNA does not code for a protein, but instead it is known it has been found that there are 3 bases which code for an amino acid. So, once you have these 3 bases such as UUU or CAC or GGC and so on, each of these codes are going to code for a single amino acid, one or more amino acids. Now, if you look at the combinations, there are 4 base pairs, and so there are 3 base pairs which are coding for a particular amino acid. So, if you look at the combination, it would be 64 ways in which 4 nucleotides can be arranged in sets of 3. Now, since there are only 20 amino acids, this is more than enough for the task required. Now, let us look at the tertiary structure of DNA. So, if one were to take a DNA and extend the length of the strand, it is impossible for us to fit this in a cell. Okay. So, therefore, what happens is that it sort of coils into a more compact 3-dimensional shape, which can fit into the nucleus. This process is known as supercoiling. And of course, needless to say, the tertiary structure is often neglected or ignored, but it is important to the action of several drugs. Okay. Now, how does this supercoiling take place? What happens is that it uses an enzyme known as a family of enzymes called as topoisomerases, and these can catalyze the act of passing one stretch of DNA double helix across another. The mechanism by which this happens is that they cleave one or both of the strands and create a temporary gap, and then once the crossover has taken place, they reseal the strands. Okay. What happens is that supercoiling allows for efficient storage of DNA, but keep in mind that the DNA has to be uncoiled again if replication and transcription are to take place. Okay. The way in which this topoisomerase works is it has a tyrosine, which has a free alcohol or a phenol, which then attacks phosphodiester bond and kicks out the alcohol, and what it does is it forms a temporary covalent adduct with DNA, which is then subsequently reacts with a proximal DNA and forms the other bond. Okay. What happens is a temporary covalent bond between the enzyme and the 5' end of each strand is formed, and once this is formed, the strands are now pulled in opposite directions, so that there can be a gap through which the other DNA can be passed. Okay. So, this is a very systematic way of supercoiling, and this after this is done, the nicks are then resealed by the enzyme. Topoisomerase 1 is very similar to topoisomerase 2 in that it relieves this torsional stress of supercoiled DNA during replication, transcription and repair. So, this stress that the supercoiled form is obviously not useful when we are doing directly. If you do replication directly from the supercoiled form, it is not going to be useful. So, therefore, the stress has to be relieved, and then only then the replication and transcription can occur. Now, the difference between topoisomerase 1 and topoisomerase 2 is that it cleaves only one strand of DNA, whereas topoisomerase 2 cleaves both strands of DNA. There is another isoform which is called as topoisomerase 4, which is a bacterial enzyme, and it carries out the same process in bacteria and is a very important target for fluoroquinolone antibacterial agents. We shall look at fluoroquinolone antibacterial agents later in the course. So, DNA is not an isolated macromolecule within the nucleus of the cell. Okay. It is associated with a variety of proteins such as histones in a structure called as chromatin. So, as we just saw, this is the primary structure of DNA, where you have a sequence which can then fold into a secondary structure, which is the double helix as shown here, and then this double helix can be coiled and supercoiled, and once it can then be associated with proteins such as histones, and it is then further packaged into histones and chromatin. The histones and associated DNA form a structure called as a nucleosome, which occurs regularly along the length of the chromatin and plays a crucial role in the regulation of DNA transcription. We shall look into some of these concepts later in the course. Okay. Lastly, we are going to look at genetic polymorphism. So, once this replication occurs, that is a DNA double helix forms its dot DNA double helices, occasionally this replication does not occur to 100% efficiency. That means that there can be a mutation that occurs. If this mutation is fatal, that means the cell does not survive because of it, then obviously it is not carried over to the next generation. But if the mutation is not fatal, then it can be carried from generation to generation. what happens is that this leads to different individuals having slightly different gene sequences. Now, this becomes important because when we are administering drugs to individuals, each individual can actually have a slightly different reaction. On an average, there is a difference of 1 base pair every 1000 base pairs between individuals. Now, what, as I mentioned earlier, this difference in sequence translates to a difference in the amino acid code or the primary structure of amino acids. So, any single change in the sequence of DNA can result in a different amino acid being incorporated into the protein. This single amino acid change may or may not have an effect on the protein's structure or function, but it is important that this difference can manifest itself in when it is interacting with certain drugs. Genetic polymorphism has very profound consequences in the susceptibility of individuals to disease and also to therapy. So, now there is an effort that is being made to incorporate these types of concepts when we are treating individuals. This is known as personalized medicine. So, if the sequence of the individual is known and if you are able to map out some of these differences that occur, and if you are able to predict or understand how these sequences or these differences in the protein structure can manifest itself during treatment, then it is possible to be able to avoid fatalities if a person reacts negatively, or to be able to administer a drug which can be more efficacious to the individual. So, this entire field of personalized medicine is now emerging.